So now that we already understand the concepts of natural selection and heritable adaptive traits and relative fitness and alleles and so on, let's start talking about microevolutionary forces. We've already been introduced slightly to the idea of microevolution. So let's see how is it happening. So small steps of evolution can happen or need these particular events in order to happen. And those are, as you can see, mutation or gene flow or genetic drift, natural selection, and a part of natural selection, sexual selection. So please take a moment, turn the video off, take a moment and think and come up with an explanation or of an idea what each of these terms refer to. And every time you learn new concepts, examples are very useful to understanding these concepts. And yes, it might seem difficult, but everything you ever did was difficult initially. But if you're very highly motivated, you put the time into it, and now it's a piece of cake, right? Or a walk in the park, or whatever you want to call it. Probably the walk in the park is much healthier. Go for that. So mutations. The word in Latin refers to change, and what exactly changes from what to what. So again, take a moment and write down or verbalize in front of the mirror with a friend and so on. What exactly is your understanding of this concept of mutation? And as I said, you always need examples. And in this case, you will soon learn that there is a variety of types of mutations because things changed in your genome or the entire genetic makeup in all of your cells at different levels, at the gene level, at the chromosomal level, and so on. So according to those levels of change, mutations can have different types. But first of all, mutations literally refer to any change in your genomic sequence. And the genomic sequence refers to all the nucleotides in all your genes, in all your chromosomes, and so on. Those structural modifications or changes within the gene will lead to a different amino acid in a particular protein, and that can lead, doesn't always do so, but it can lead to phenotypical or physical modifications that in particular in that individual. And that can affect how natural selection perceives that individual, more fit or worse fit, or completely unviable. And in that case, we can talk about lethal mutation. So speak about types of mutations that you already reviewed and looked into it and tried to come up with the ideas that you already have and know about the concept of mutation. They could happen in the sequence, the genomic sequence, in just one location. And because it's just one pair of nucleotides that is different or changed comparative to the copy of the DNA that this new DNA was copied from. And so it's just one pair of one pair of nucleotides. If those two nucleotides that changed are the same size, so it's a purine to a purine, or purine to a purine, or a pyrimidine to a pyrimidine, we call that transition. And if it is from a pyrimidine to a purine, so from A to C, let's say, or from A to T, we call that a transition. Notice that this is actually a link, and it will send you to a very clear picture that shows you, compares and contrasts the concepts of point mutation within between purines or in between or transition from purine to pyrimidine, and gives a lot of examples. Now, according to how natural selection sees this mutation, according to what will happen to the protein that gene that was changed codes for, how would that protein or the sequence of amino acids will be changed, how dramatic would be change, that would be deem that mutation a silent mutation, a missense or nonsense. So again, take a minute, turn the video off, and try to speculate from what you already know. How are these type mutations affecting phenotype? And of course, definitely resource to your textbook. And as you can see, there are also links. So a silent mutation, as you very well speculated, would leave the amino acid sequence or the primary structure of the protein that gene that mutated or changed unchanged. So that means that even though the nucleotide changed, due to the degeneracy of the genetic code, because we know there are only about 20 amino acids, but there are 64 codons coding for each of these 20 amino acids, it means that there are more codons coding for each independent individual of these 20 amino acids, meaning that even though the nucleotide could have changed, it would still be a very similar codon coding for the same amino acid, leaving the sequence of amino acids in the primary structure of protein unchanged. So that means it's not even visible to the natural selection because nothing has changed in the phenotype. Now, if that codon has changed enough that it doesn't fit in that group of 2 to 6 codons that code for the same amino acid, that means the protein is changed. And that means that probably that protein cannot perform the function that was adapted to perform, and we call that type of point mutation a missense. When the codon is changed from a coding codon to a particular amino acid to one of the 3 stop codons, then that means the protein would be incomplete, and that type of point mutation is called a nonsense point mutation. These mutations, so in the case that we just discussed, the point mutations of transition transversion means that one nucleotide has been replaced by another one. But the rest of the codons are the same. Now, if just one, could be a lot more, but if one pair of nucleotides has been deleted or an extra one has been inserted, that means that down the line of that eventually messenger RNA sequence would be completely shifted, and that's how we call these mutations frame shift mutations, and usually they lead to a completely unfunctional protein. A very important type of mutation that you already read extensively in your additional reading, Making of the Fittest, which brings the ability of allowing for further mutation without losing a function and maybe gaining a function. It's this concept of gene duplications. So again, take a moment, turn the video off, and try to remind yourself of what type of proteins these gene duplications have allowed, for example, the bloodless fish or ice fish of the Bouvet Islands to acquire from gene duplication. And keep this concept in mind. You will find it in future chapters of this great read of Making of the Fittest. Sometimes entire pieces of a chromosome can be moved either within that same chromosome or from one chromosome to another. Which reminds me, I would like you to also try to test yourself in between these concepts of what is a gene and what is a chromosome. Are there more genes on a chromosome, or are there more chromosomes on a gene? So explain it to yourself, putting in writing, it helps tremendously to verbalize these concepts. And drawing helps as well. Sometimes a piece of chromosome can be moved, so that's translocation, or a piece of chromosome can be just turned upside down or upside right, depending how you look at it. It's all a sphere, it's moving around. So just the sequence of the genes within a chromosome can be inverted. And the sequence of the gene on the chromosome is very important to how they are read. So biochemical pathways rely on a sequence of genes that control the production of a particular succession of enzymes that can produce an end product from an original very different product. Somatic mutations are not so important to the understanding of evolution because they refer only to the soma or to the body, and they are acquired through your life, and they are thus not inherited. They're still important to look into it. They're definitely a type of mutation, but they will not affect necessarily future offsprings. They could affect the individual that has somatic mutations, fitness level, but not necessarily future generations. And speak about future generations, depending on how these mutations are changing the proteins that they are, the gene that mutated it's coding for, natural selection can deem those mutations beneficial because they give an advantage, an adaptive, heritable trait, or more often than not, a harmful effect, and we also call that deleterious because it could lead to a lethal allele, which means it deletes the individual that carries that particular mutation. But most of the times mutations are simply neutral, so that means they neither give an advantage or a disadvantage, and all of us are mutants, and all of us carry almost 200 different mutations that can affect us in a way or in a beneficial way, hopefully, or they're not detrimental enough to kill us. This is an interesting example, and different versions of your textbook have taken the time to look into various concepts of superbugs, in this case super rats, and so on. So this, I find this as an interesting story from referring to a chemical that is naturally produced by fungi on plants such as grasses, but also legumes. And this particular compound prevents coagulation, and was first as a very big problem in cattle farming in northern United States and southern Canada, so in states such as Wisconsin and Alberta, sometime in 1920s. Cattle that either performed or were exposed to a minor surgery have bled to death, or they just had internal hemorrhages that killed them. And of course farmers were distraught. What is going on? What is happening? Luckily, biochemist came to the rescue, and he put all his other research on the side to take a look at what is happening in those cattle. And they found out that it turns out that rotten sweet clover, so a particular legume that was brought from Eurasia, which is now an invasive species, so sweet clover could be white sweet clover or yellow sweet clover, very common weed, unfortunately, but nonetheless a legume, which is important for animal feed because it has the ability to have endosymbiotic bacteria in their roots that can transform otherwise inaccessible atmospheric nitrogen into nitrogen that the plant can use to make its own proteins and DNA. So nitrogen is a very important limiting source of and protein production. So it's very important in nature. And legumes cannot do it themselves, but they adapted to facilitate symbiotic relationships with bacteria that can in their roots. So thus, legumes are very important to farmers who want their feed to be highly nutritious. Unfortunately, in this particular case, maybe because of a much rainier season, those that forage, that hay has rotten, and the fungi transform the chemistry of that feed, and unfortunately was rather killing the cattle instead of helping them grow stronger. So from that, today, we have several different compounds that includes coumarin and coumadin, which we know today are used to reduce chances of clotting for people that already had a stroke or a heart attack. Originally, it was only developed into warfarin, which happens to be a rodenticide.